The diencephalon is one of the four major components of the brain, occupying an unpaired middle position. It is connected caudally to the mesencephalon and is located at the base of the cranial cavity, mostly surrounded by the telencephalic hemispheres. The diencephalon consists of five different structures: the thalamus, which is the largest part, the epithalamus, located superiorly to the thalamus, the subthalamus and hypothalamus, located anteriorly and inferiorly to the thalamus, and the metathalamus, made up of nuclei attached to the thalamus in its posterior region, medial and lateral geniculate nuclei, arranged bilaterally. This last structure can be considered part of the thalamus. The diencephalon is almost completely hidden by the telencephalic hemispheres, except for its ventral part, where the hypothalamus is located. In order to study its position and the relations it builds with the surrounding structures, it is essential to largely remove the telencephalic hemispheres. The shape of the diencephalon can be roughly compared to a truncated pyramid, compressed on each side. Its anteroposterior diameter is significantly larger than its vertical and transverse diameters. As we mentioned before, the diencephalon is located cranially to the brainstem, forming an obtuse angle open forward and downward, due to the forward tilt of the brainstem. The ventral or hypothalamic surface is the only visible part of the diencephalon. It lies in contact posteriorly with the brainstem, anteriorly with the optic chiasm and the two optic nerves cross, which separate it from the frontal lobes, and laterally with the temporal lobes. The hypothalamic surface also rests on the sella turcica of the sphenoid bone. Coming into contact with arteries, they make up the circle of Willis and with the venous sinuses of the cranial base. When observing the external configuration of the brain from its underside, the diencephalon structure visible in the anteroposterior direction are the optic chiasm, a formation resulting from the partial crossing of the optic nerves, the tuberum cinereum, which extends downward like a small funnel, giving rise to the infundibulum. The infundibulum is continuous with the stalk of the pituitary gland. The mammary bodies, namely symmetric projections located in front of the interpeduncular fossa, cerebral peduncles. The subthalamus, located in the inferior part of the diencephalon, serves as a transition area between the mesencephalon and the diencephalon. However, being a boundary area, it has no clear demarcation limits. The superior surface of the diencephalon forms the roof of the third ventricle cavity. This cavity is one of the communicating cavities in the brain containing cerebrospinal fluid and is laterally bordered by the medial portion of the right and left thalamus. The third ventricle is so large that the diencephalon can be described as the collection of structures surrounding this cavity. The superior surface is covered by the choroid tela, a thin triangular membrane that, when folding upon contact with the diencephalon, contributes to the formation of the lateral ventricles. We will explain its functions in the next videos. In the posterior portion of the superior surface, we find the habenula, a fibrous cord that medially limits the epithalamus with its attached nuclei, situated in a triangular region called the habenular trigone. The habenular trigones extend backward to the pineal gland, the primary structure of the epithalamus. Each lateral surface of the diencephalon consists of a symmetrical gray mass ovoid in shape, the thalamus. The thalamus is positioned obliquely, tilted forward and inward, with its anterior poles much closer than the posterior ones. As previously mentioned, the medial face of the two thalami enclose the cavity of the third ventricle. Laterally, the thalamus comes into contact with the telencephalon through the interposition of the internal capsule. The division between the thalamus and the telencephalon, however, is purely conventional. as the gray matter of these two structures is not sharply separated. The anterior surface is very narrow and includes only the anterior poles of the right and left thalami. It is bounded inferiorly by the optic chiasm and superiorly by the interventricular foramina of Monro, which allow communication between the third ventricle and the lateral ventricles. The posterior surface can be identified as a virtual plane extending from the posterior commissure, or white matter cord belonging to the pineal gland, to the mammary bodies of the hypothalamus. This area is traversed by the upper part of the Sylvian aqueduct, which connects the third and the fourth ventricles. The subthalamus is also located in this region. The thalamus is an ovoid structure approximately 4 cm in size and represents the largest component of the diencephalon. It consists of a collection of nuclei encased in white matter, from which fibers project to the semi-oval center of the telencephalon. Thalamic nuclei are classified as either specific or non-specific, depending on whether the efferent fibers project to well-defined areas or broader regions of the cortex. Non-specific nuclei, in particular, are involved in regulating alertness. The thalamus occupies a central position in the brain and establishes close relations with the adjacent structures, so much so that it is sometimes referred to as the thalamencephalon. Its primary function is to relay ascending tracts from the spinal cord and brainstem to the cerebral cortex. Additionally, it transmits messages processed by subcortical circuits, such as those from the basal nuclei, to the cortex, and facilitates cortical interconnections. The function we first mentioned, called relay, is essential for tactile, thermal, pain, and proprioceptive sensations, as well as for specific sensory pathways such as taste, vision, and hearing. In summary, the thalamus is a complex structure with a central role in different brain functions, acting as a hub for processing and distributing information between the external world and the cerebral cortex. The thalamus is conventionally divided into 4 surfaces and 2 poles. Superior, dorsal surface. It consists of 2 triangular regions, one medial with a posterior base and one lateral with an anterior base, separated by the choroidal fissure. The medial region is covered by the ependema, the membrane forming the floor of the lateral ventricle. The lateral region is covered by the thala choroidea and extends posteriorly into a globular enlargement known as the pulvinar. The boundary between the medial and superior surfaces of the thalamus is marked by the stria medullaris, a white matter tract to which the membrane of the third ventricle is attached. Medial surface. This surface forms the walls of the third ventricle and is therefore lined by ependema. It is often connected to the medial surface of the contralateral thalamus through the interthalamic adhesion, a bridge of gray matter. The medial surface includes a layer of white matter and its upper portion, known as the zonal layer, which is covered with a gray matter layer known as the periventricular gray matter. Inferiorly, the thalamus is separated from the subthalamus and the hypothalamus by the hypothalamic sulcus. Lateral surface. The lateral surface contains the reticular nuclei and is related to the internal capsule and the lenticular fasciculus. White matter structures we will discuss later. Inferior ventral surface. The inferior surface does not have a distinguishable surface. Anteriorly, it corresponds to the hypothalamus, while posterolaterally it corresponds to the subthalamus. Anterior pole. The anterior pole is located in the depression formed by the head of the caudate nucleus and is related to the concave part of the fornix columns. The relationship between the anterior pole of the thalamus and the fornix columns delimits the foramen of Monro. Posterior pole. The posterior pole corresponds to the pulvinar, the largest nucleus of the thalamus. Below it lie the medial and lateral geniculate bodies of the metathalamus. As we mentioned earlier, numerous efferent fibers emerge from the thalamus. In some areas, these fibers form thickened bundles of white matter known as the thalamocortical radiations. We distinguish 4 types of radiations: anterior, frontal, connecting the anteromedial part of the thalamus to the frontal cortex; posterior, occipital, connecting the lateral geniculate body of the metathalamus and the lateral surface of the thalamus to the occipital lobe and part of the parietal lobe; superior, parietal, connecting the vetrolateral part of the thalamus to the precentral and postcentral gyri; inferior, temporal, connecting the medial geniculate body of the metathalamus and the posterior part of the thalamus to the temporal lobe. Cortical projection to the thalamus are exactly reciprocal. Each cortical area projects to all sides of the thalamus, from which it receives stimuli, following a topographically organized manner. The thalamus is entirely enveloped in a thin layer of white matter known as the external medullary lamina. Laterally, it separates the reticular nuclei from the rest of the thalamus. It then penetrates the gray matter of the thalamus to form the internal medullary lamina, which subdivides the thalamus into the various nuclei. Focusing on the course of this lamina will allow us to identify the delineation of the nuclei. The internal medullary lamina follows a median path, but bifurcates into a Y shape as it progresses toward the anterior pole. This bifurcation is important for the division of nuclei into medial and larger groups. Thalamic nuclei are classified according to 2 main criteria: topographical and phylogenetic. First, let's consider the topographical classification, based on the course of the internal medullary lamina. The median course of the lamina allows us to separate a medial nuclear group from a lateral nuclear group. The lateral nuclear group is further divided into dorsal and ventral groups by a thin white matter lamina. The anterior nuclei are found at the bifurcation of the internal medullary lamina. Finally, another group of nuclei, the intralaminar nuclei, are embedded with the internal medullary lamina. These are often grouped together with the midline nuclei, found on the surface of the third ventricle beneath the ependyma. According to the phylogenetic classification, there is an older group of nuclei, known as the paleothalamus or allothalamus, consisting of the intralaminar and midline nuclei, and the more recent group, the neothalamus or isothalamus, which comprises all the remaining nuclear groups. The reticular nuclei are referred to as the perithalamus. With the exception of the reticular nuclei, all other thalamic nuclei are connected to the telencephalic cortex. Based on their efferent connections, we can divide them into 3 types. Relay nuclei, which can further be divided into specific projection nuclei that innervate a specific cortical area, and diffuse projection nuclei that innervate a broader area. Intralaminar and midline nuclei, which make up the allothalamus. These function as diffuse projection nuclei. Reticular nuclei, which have no cortical projections. It is useful to delve into the physiology of thalamic nuclear groups, especially the relay nuclei, in order to define the type of information they convey. Anterior nuclei, important stations for integrating the endocrine functions of the hypothalamus with the limbic lobe of the cerebral cortex, turning vegetative information into experience and memory. Ventral lateral complex transmits motor information to the primary motor area and the prefrontal cortex. Ventrobasal nuclei transmits sensory information to the primary sensory area. Pulvinar belongs to the dorsal nuclei, is the largest nucleus, and plays an important role in attention processes triggered by visual stimuli. Mediodorsal nucleus involved in working memory and experiences related to the olfactory stimuli. Geniculate nuclei, also sensory in function, with the lateral geniculate body projecting to the primary visual area and the medial geniculate body projecting to the primary auditory area.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.